on, I have to let one more person in. So I totally get it if you don't have these with you, or if you don't have the page that has all the stuff that we need to know for the test on it. I picked out, as usual, the slides that I would have used in class, and I did extensively abbreviate the beginning part of this chapter. Unfortunately, in this context, I am not interested that you know about all of the experiments that each person did and how they figured out both that messenger RNA actually coded for amino acids, and how they figured out the messenger RNA codons that make up the chart. So we're just going to skip that interesting part and just talk about the essential parts. I would remind you, I put this in as the first slide to remind you that these are the 20 amino acids. What they all share in common is pictured here across the top, right? An amino group, a carbon, a carboxyl group, a hydrogen, and then that R group or functional group that is the side chain. And different amino acids, that's what makes the different amino acids have their chemical tendencies. And you'll notice they're put in groups here: non-polar, aromatic, positively charged. And that is why we have 20 different amino acids, because there are 20 different of these side chains or R groups. This is a larger diagram reminding us about how a peptide bond is formed, because that's how we make a protein, is by stringing these amino acids together into a polypeptide first and then into a protein. Notice that water comes out, so it's a dehydration synthesis reaction, and we're going to form a chemical bond here between the carbon of the carboxyl group and the nitrogen of the next amino acid. Proteins have levels of organization or levels of structure. The primary structure we refer to as the list of the amino acids in order from amino acid 1 to however many there are. Most polypeptides that are going to form proteins or subunits of a protein usually have between 100 and 200, say, amino acids. These R groups of the primary amino acid chain are going to interact chemically to form secondary structure. And those of you that remember this from chemistry or bio, you might remember that there's two different forms of secondary structure: the alpha helix or the beta pleated sheet. They're showing you alpha helix here. Tertiary structure is when this secondary structure, when R groups on the secondary structure start interacting and forming chemical bonds that like a ionic bond, or maybe a disulfide bond, or maybe a covalent bond. And you can see a subunit. This looks like hemoglobin to me. It looks like the iron binding unit there, the part that binds the oxygen rather, right? So this is tertiary structure, when we have the secondary structures sort of form a globular protein. And then quaternary structure, when you have more than one of these subunits, or sometimes you'll see them called domains. That's the more modern term. We used to call them subunits, but you'll see more in textbooks now, the word domain. And this is hemoglobin, and you can see there are 4 domains to this particular protein. Each one of them has its own primary structure, secondary structure, tertiary structure. Okay. But the molecule is only functional when you put all 4 of them together. So that's to remind you about protein structure. Now we're going to go back and just focus on this primary structure of amino acids and how that gets built inside cells. So that's going to happen in what we call translation. And for that, we're going to need transfer RNAs, ribosomes, and the message itself. For the exam, I wanted to tell you this now while I was thinking of it. For the exam, you are going to need to have a copy of this code with you. I don't know how to put one on the test. So you're going to have a copy of the chart with you, because I will ask you some questions some serious, easy gimme questions. If this is the messenger RNA, what is this primary sequence of the amino acids in the polypeptide or in the protein? Okay. By now, you should all already know how to read this chart, but if you need a little refresher, remember for messenger RNA, every codon, I should probably add that to this transparency right now, right? A messenger RNA codon is 3 bases or 3 nucleotides equals a codon. And the way that you read this chart, you can only read the message from this chart, and you would look at the first base, and every chart you're ever going to see is always in the same order. It's always UCAG, and then across the top UCAG. So for example, if we were looking for the messenger RNA codon CCU, and we wanted to know what amino acid that coded for, we would look at the first base here is C, second base C, and we would bring our fingers together and say, oh, CCC is proline. Okay. The chart is only going to give you the 3-letter code, and some charts actually will only give you a single-letter code, which you'll see if I go back to that first transparency that I showed you about the different amino acids, right? So if you look at proline in particular, if I can find that one, that's an easy one because the 3-letter code is pro and the 1-letter code is just P. So again, you guys are going to need to have a copy of this messenger RNA codon chart with you for the exam. All right, so now on to translation. Okay, the 3 things that we need, remember we need transfer RNAs, we need a ribosome, and we're going to need a message. This next section talks about the transfer RNAs and how they get chemically prepared to participate in translation. Each transfer RNA has the same sequence at its 3' end. It always is CCA, and they're showing you a blow up of the adenine. So they're just putting CC and then this blow up of the adenine, and they're showing you the chemical bond to add a particular amino acid. All right. This process requires an ATP in order to charge the transfer RNA. Okay, this is important because we're going to count this. So if I would ask you a question, how many ATPs would be needed to translate a messenger RNA sequence that had 89 codons, you would have to add 89 more ATP molecules because each of the charging of each of the transfer RNAs counts as an ATP. Okay, we'll see that translation itself, both releasing the elongation factors and the peptide bond formation and the translocation of the ribosome, that is also going to cost the cell 2 more energy molecules. There are 20 different enzymes that are unique, one for each of the amino acids. Those enzymes are called amino acyl transfer RNA synthetases. And as I said, they are separate amino enzymes. There's one for each of the 20 amino acids. Okay. They live out in the cytoplasm in a eukaryotic cell, and they, of course, are involved in charging and recharging these transfer RNAs after they deliver their amino acid in the ribosome. All right, so let's look at translation itself. It has 3 parts: initiation, elongation, and termination. Okay, and your book has this divided out into 7 steps. I would know them. Like I said, I realized I posted the handout super late at like 5:20. We had a department meeting today, so I was like, oh, I have plenty of time to do that. And then suddenly I had no time to do that. So when you look at the handout, you'll see that I put there the specifics and the things, the answers to the questions that you'll need to know in order to be able to answer the questions in the test. So I would remind you, this is a bacterial ribosome. Small subunit is 30S, large subunit is 50S. Eukaryotic ribosomes are a little larger and more dense. We have a 40S subunit and a 60S subunit. What helps orient the messenger RNA correctly into that small subunit, or helps the message bind to the small subunit of the ribosome, is this particular sequence. It's called the Shine-Dalgarno sequence. And I'll blow it up for you here. It actually binds to the 16S ribosomal RNA, which you might remember is part of the small subunit of the ribosome. We talked about that very briefly last time. And this is the sequence. Okay. And this sequence is, of course, complementary to the ribosomal RNA, and that is going to help attach and orient the message so that it's in the right spot for translation to occur. Notice the initiation codon here, the AUG, is a little bit, we would say, downstream of the Shine-Dalgarno sequence. Okay, so we're in a bacteria. So here's the messenger RNA binding to the small subunit of the ribosome. Okay, it's going to bind so that the Shine-Dalgarno sequence is kind of over the E site, what will be the E site, and that AUG, the initiator codon, is going to be lined up with the P site when the large subunit comes down. Notice there's an IF3, that stands for initiation factor 3. It binds that small subunit of the ribosome, and it actually prevents the large subunit from joining until the message is aligned, and as we will see, the first transfer RNA is in position. Okay. So message attaches to the small subunit of the ribosome. The Shine-Dalgarno helps with the orientation. The initiation factor 3 prevents the large subunit from binding too soon. The first transfer RNA that has the formalin or formilated methionine that's going to match its anticodon perfectly to the first codon of the message is going to bind to the message. Notice also at this time, initiation factor 2 joins. So this forms a complex. So the transfer RNA forms a complex with the IF2. And notice this is going to require a little bit of energy. 1 ATP or GTP. They're the same, it's just a different base here. Instead of adenine, it has guanine. Notice this, another initiation factor is going to also join to the bottom of the small subunit of the ribosome. Now that all of this is in place, the GTP is going to be used. It's going to detach all three of those initiation factors, and the large subunit is going to plop down on top. Okay. At the end of initiation, the E site in the ribosome is empty still. The P site has that first transfer RNA in it with the formilated methionine. Now, AUG might be used later in the protein synthesis, but it will never have the formal methionine on it again. It will just be a regular methionine. Okay, before we talk about elongation, that's just to remind you that that's sitting in the P site. Before we talk about elongation, I did want to bring up the difference in eukaryotes for initiation. Okay, it's a slightly different sequence. It's called Kozak sequence, and notice it's not upstream of the first AUG start codon. It's the AUG start codon is a part of this sequence, so it's not exactly homologous to the Shine-Dalgarno. We would say it's analogous. This Kozak sequence is going to also contribute with aligning the message in the small subunit of the ribosome, but look, it's a lot more complicated in eukaryotes. Other parts of the post-transcriptional modifications that we talked about last time, including the 5' cap, a protein that binds to that's called the cap binding proteins, or CBP for short, and you can see there's 2 of them here, as well as the poly A tail, are all involved helping to align the message in the right spot in the ribosome and to stabilize the ribosome. Okay, notice the ribosome is positioned here a little bit before the first before the start codon here. So it's a little different than in prokaryotes. But like usual, because we know prokaryotes better, we always use that for our example. Okay. So we've already discussed the initiation, right? But I wanted to just remind you again that the ribosome has 3 places, 3 holes, 3 pockets or grooves where transfer RNA molecules can fit in. And as we're transitioning from initiation to elongation, that's the P site is occupied by this first transfer RNA with the formulated methionine on it. Okay, now step 2, bio 1, and in microbiology, we just said the second transfer RNA fills the A site, right? A little more complicated than that, so we're adding a little bit more of the complexity. This transitions us to what we would call elongation, and the proteins that assist in elongation, they're going to be called EF for elongation factors, and there are a couple of them. This one here, it's called EFTU. EFTU is going to complex with another GTP, and they are going to guide that second transfer RNA in to fill the A site. Okay, so it doesn't just wander in there by itself. It has help. Okay, so the elongation factor 2, it's actually called 2, with the GTP, that's going to fill the A site. Okay, there it is. They popped in there. After the charge transfer RNA is filled in the A site, the GTP is cleaved, so we're going to release the energy, and that's going to be used to form the peptide bond. Okay. The elongation factor 2 and the GDP are going to exit, and this molecule here, elongation factor TS, is a helper. It grabs EFTU and it drags it along to help attach it to another GTP and to the next transfer RNA. Okay, so its job, elongation factor TS, its job is to regenerate or reconstitute EFTU so it can be used again with another GTP and another transfer RNA for the third transfer RNA that will need to go into the ribosome. There they come out, right? Peptide bond is formed, I already said that. And we both list that as step 6. Now the ribosome needs to move over 1 codon, that process we call translocation. And that is going to require a third elongation factor G, and a second GTP. So each transfer RNA that's going to add an amino acid to the chain during elongation is going to require 2 GTPs. Okay. Every transfer RNA required 1 to get itself charged with the enzyme. And so usually people say it requires 3 ATP or 3 energy molecules in order to add every amino acid to the chain. 2 for the actual process of translation, and 1 for the charging of the transfer RNAs. So after the translocation, what that means is the ribosome is going to move over 1 codon. So now the first transfer RNA is in the E site for exit. It doesn't stay there very long. It gets pushed immediately out into the cytoplasm. The second transfer RNA that has the growing chain, that's in the P site, and the A site is ready to accept another transfer RNA with the correct anticodon. This continues for however many, 100, 120, 85, however many amino acids this particular message codes for. And then the third part of translation is termination part, and that is also going to require proteins to help release the peptide and to open up the ribosome again so it can take on another messenger RNA. And those factors are going to be called, just like we had IF for initiation factor, and EF for elongation factor, these factors are going to be called RF for release factors. And I think I put it all together on one. I just took the one that has them all on the same slide. So when the ribosome translocates to a stop codon, there's no transfer RNA that matches that stop codon. So that is going to trigger RF1 and RF3. RF1 actually fills the A site. Notice RF3 is going to grab a GTP. This is going to cut off the polypeptide. 1, 2, 3. And then all of this kind of happens at once. The GTP associated is hydrolyzed. The ribosome is going to come apart or be dissociated. And release factor 1 here is going to be let go, right? And so is this last transfer RNA that would have been sitting here in the P site. Release factor 2, actually, I think complexes with 1. So I know I said there were 3, and what I wrote down in my notes was RF1 and RF2 actually fill the A site, and I think RF2 is the one that's involved in cleaving the polypeptide. RF3 causes the ribosome to change shape and separates the 2 subunits. I put this chart in because it's a quick summary of everything that we just went through for each of the steps: charging the transfer RNAs, initiation, elongation, and termination. And then the last one I included was a 3-dimensional picture of what the ribosome actually looks like. And I thought this was kind of cool because these are 3 transfer RNAs that were in this particular ribosome in the E, in the P, and the A sites. Right, you can see the polypeptide chain growing off the top, and you can actually see how the message lays in the small subunit of the ribosome. About 9 of the, let's say 9 bases, right, or 3 codons are going to be available on the small subunit of the ribosome at a time that align with the E site, the P site, and the A site. Okay. That ends